Come on. Can somebody confirm you can hear me?
I can hear you.
Thank you. Let me get there. Not sure what chapter we're on. Take a look here. I share my screen as soon as I blow that up. All right, you should be seeing my screen now. Today is June 11th, and we should be on chapter 7. I don't think we're quite there yet. Let me take a look at chapter 13. Come on. We talked about latent and persistent viral infections last, so we have a couple more slides of chapter 13. My voice is hoarse again. All right, any questions before we begin? Let me state that I've done some grading. I think I've done all of the labs now 2 times. I believe I've graded the worksheet to lecture 13 or chapter 13. And I think I have all the extra credit assignments graded. Let me state that the unknown notes page, let me show you the syllabus here. The unknown notes page is ending on Friday, June 14th, meaning that's the last day that unknown notes page will be open. If you request a test on that day, it must be a 1-day test. There are a few, but most of the tests are 2 days. So if you have a 2-day test, you have to request it on Thursday. I was surprised that a number of students requested a test today, and I thought most of you were done. You need to finish soon because the unknown notes page will be closing on the 14th, Friday the 14th. And then the project must be turned in on Saturday, June 11th. Excuse me, June 15th. Can't read this. Let me pull it up a little bit and clean my glasses. But the unknown notes page will be graded. So make sure you find out what that assignment is supposed to have in it. Meaning make sure you have all of the topics to get all of the points. A lot of you have not put up your last test request. And to get full points, you must have the last test request up there. So a lot of you need to add to your unknown notes. This will be my one warning for you to finish your unknown notes page. It is going to count like a regular lab assignment. So the unknown notes page will be worth 5 points. Any question about any of that?
I have a question about the upcoming final.
Uh-huh.
I see that it's due on June 19th. That is a holiday and the campus is closed. Would you be willing to let us finish that on the 20th?
The 19th is a holiday?
Yeah, it's Juneteenth and the campus is closed.
Let me get back to you on that. I'm surprised because I looked this schedule up and I didn't notice that the Wednesday was a holiday.
I believe it maybe is a newer holiday.
Okay. I've never heard of Juneteenth. Is that, no, that's not when the blacks were liberated. Anyways, I don't know, so I'll have to get back to you on that.
Okay,
Let me get back to you on that. All right. And even though the campus is closed, we are an online class, and so it's not necessary that we will be closed. But this date has been scheduled for the beginning of the term, so I'd hate to change it, but I will look into it. Okay.
thank you. Appreciate that.
All right. Um, there is no lab today. I will be logging in for the first 15 minutes just to answer any questions. After 15 minutes, I'll be logging out in the regular lab time. If there's no further questions, let's finish up chapter 13 because we need to get started on chapter 7. And I would like to be finished with chapter 7 today. And then on Thursday, I'll have all the day for reviewing for the final. Do note that there is a worksheet to chapter 7. It is due on Thursday, 6/8, because of the end of the term. This worksheet for chapter 7 is extra credit. So you do not have to do it. You do not get penalized if you do not do it, but you will get extra credit if you do turn in the worksheet for chapter 7. And I think the extra credit is the same as no, because it's 2.5 points. So I think it's 2 points for the worksheet, but it states on the assignment what it is. Maybe I can find that quickly here. Not there anymore. I say 1 or 2 points of extra credit. All right, any question about any of that? If not, let's go to the chapter 13. So I need to talk about prions. Prions are a protein which is infectious, and they actually call it a proteinaceous infectious particle that causes disease. There are no other proteins that cause disease. It is an infectious protein. It can be inherited and it can be transmissible by ingestion, organ transplant, or by transplant by surgical instruments. It does cause spongiform encephalopathy. This is vacuoles in the brain in humans and other animals. Let me show you a picture of that, which I should actually put that over here. There we go. Blow this slide up. So this is a slide of a normal human brain. This is a slide of a Kuru patient. This is a slide of classic Creutzfeldt-Jakob disease. And this is a slide, this is probably of a sheep because scabies is a disease of sheep. But spongiform encephalopathy because the brains sort of look like sponges. Any question about any of that? And that's where the spongiform name comes from. In animals, prions can cause bovine spongiform encephalopathy, which you may have not ever heard of, but I bet everyone's heard of mad cow disease. That's the common name for it. It also can cause scabies. This is a disease in sheep that European sheep ranchers have been living with for centuries. and cervid chronic wasting disease. It can also cause, it's a disease in, I think it's elk in Colorado or something like that. In humans, we don't call it things like that. We call it Creutzfeldt-Jakob disease after two doctors who recognized it in a family, or Gerstmann, Strassler, Schinker syndrome, who were three doctors who recognized it in another family, and fatal familial insomnia and Kuru. These are the human diseases, which are a prion disease, meaning a proteinaceous infectious particle. Kuru is perhaps the best known, and it was a disease seen in the Fore tribe of Papua New Guinea. Here, the red of Papua New Guinea is where the Fore tribe is the people of Papua New Guinea and the Fore tribe. When you got Kuru, the patient became unable to walk, loss of the ability to swallow or chew, drastic weight loss, and then it led to death. How was it transmitted among the Fore tribe? The Fore people had an unusual funeral ritual where when somebody died, the person was cooked and then their relatives ate the dead body. Any question about that? It turns out that prions tend to get in nervous tissue, and especially the brain has a rich source of prions. And so if you eat the brain or the spinal cord, or actually any nerves, you can get the prions. And the Fore people were getting it from their dead relatives, who some person in the Fore tribe got the prion disease, and then they ate the person, and so it started spreading among the Fore tribe. Fortunately, in the Fore tribe, the mothers noticed that people, including their children, were getting Kuru, and they suspected it was from eating their dead relatives. And so the mothers stopped eating their dead relatives and then refused to allow their children to eat their dead relatives. So that when the whites came to the Fore tribe, it was only the men who were getting Kuru because they continued the practice of eating their dead. And then the missionaries saw this and they thought it was just the worst thing possible, anathema, and they stamped it out. They said, no more eating your dead. It's against, I don't know, the laws of nature or God or something like that. And actually, the missionaries, which normally got criticized for not being tolerant of other cultures, they actually did a good thing for the Tor tribe because the missionaries got rid of Kuru in the Fore tribe. Any question about any of that? So there, once again, is Kuru, and all of these are spongiform encephalopathy. Well, these two are human brains. That's probably a sheep brains. And there's a normal brain. The mechanism of the disease is that there are two proteins. One protein called PRP superscript C is a normal cell surface protein on a nerve cell, which is important in the functioning of that nerve cell. And then there's the prion disease, or the prion protein, which is called PRP superscript SC, for SC standing for the Scrapes protein. And the PRP superscript SC is the protein which causes Scrapes disease, or the prion disease. And how it replicates this protein is it comes up to the normal protein, the PRPC protein, and converts this normal protein to the scrapese protein or the prion protein. The only difference between the PRP superscript C normal protein and the prion protein is the folding of the amino acids, meaning these two proteins have the same sequence of amino acids. You get that? So they're the same proteins by the sequence of amino acids, but this one is in a different folding pattern. And what it does is this protein comes up to the normal protein and changes the folding pattern of the normal protein to the prion protein. And then the abnormal prion protein starts accumulating in the brain because the brain cell, or let's say the nervous cell, recognizes that the cell needs more of the normal protein, so it transcribes more normal protein and then translates it. So transcribes and then translates it. The trouble is this cell now has two prion proteins in it, and that means it's even more likely that one of the scrapese protein is going to come up to the normal protein and then convert it to the prion protein. Well, the cell will notice that it needs more of the normal protein, so it'll transcribe and translate more of it. And that will cause the accumulation of the scrapese protein in the nervous cell, which in a brain cell, the scrapese protein will accumulate, start forming plaques, and will eventually lead to the nervous cell dying, causing the prion disease, the infectious particle. Anyways, that's how the proteins replicate themselves. Any question about any of that? If you remember, in England, they were, actually Great Britain, they were feeding cow brains to cows. What they did was they ground up the cow brains, put it on grain, and then fed the grain to cows so that the cows were eating essentially cow brains. And that caused the mad cow epidemic in Great Britain. One of the cows got prion disease, and then they ground up the brain, put it on grain, and then fed it to cows. And so it just caused more cattle in Britain to get the prion disease, the mad cow disease. Actually, about 150 British citizens, meaning people, came down with prion disease, probably from eating tainted beef. And then also about 150 British cats, both zoological cats and then kitty cats, came down with prion disease. All because of tainted British beef. Surprisingly, dogs appear to be resistant to at least the consumption of the scrapese protein, because the dogs, which were eating similar to food to the British cats, did not come down with prion disease. Any question about any of that? How the British solved this was they incinerated all of their cattle, and then they bought cattle from outside Great Britain to repopulate the cattle herds. All right, any question about prion disease? If not, let's move on to plant viruses and plant viroids. Plant viruses resemble animal viruses in many ways. They do cause many disease among plants, including diseases of economically important crops such as beans, corn, sugarcane, potatoes. Because of the tough cell wall of plants, though, the plant viruses tend to enter the plants either through a wound or via a parasite, such as a nematode, a fungus, or an insect. Infected plants can then spread the plant viruses in either their pollen or their seeds. So obviously, if a corn plant were to become infected with the virus, it could spread with an insect eating the corn plant and then spreading it to other corn plants the insect eats on. But it can also spread with the pollen or the seeds of that corn plant. Any question about plant viruses? That's it for plant viruses because obviously they don't infect humans and cause disease in humans. So that's all we're going to talk about, plant viruses. Let me talk a little bit about viroids. Viroids are a naked RNA molecule that causes disease. And the naked RNA is transmissible among organisms. Fortunately, there are no viroids known to cause any animal disease, but all viroids, at least all viroids that are known, only cause plant diseases. Any question about that? Let me see if I can get a picture from the internet. I used to have one here and somehow I lost it. Let's see, potato, spindle disease. Ah, there we go. So there's a normal potato, and there are potatoes infected with a viroid, the potato spindle disease. Any question about that? Blow it up a little bit. I should save that. There we go. Might have it in the pictures. I should look there. All right. I think that's it for chapter 13. Any questions about plant viruses, viroids, or about prions, or any animal or bacteria viruses? If not, let's go to chapter 7. I always end with chapter 7 because this is a lesson that students already are comfortable with, meaning you already know a lot about controlling microbial growth. It's going to talk about things like sterilization, disinfection, antisepsis, degerming, sanitization, autoclaving, pasteurization, biocides, and germicides. So those are the terms of this. So in this slide, I guess I should mention, I'm showing you the major goals and a rough outline. And this slide is going to largely talk about factors that can control microbial growth, meaning antiseptics and disinfectants and things like that. And students already know a lot about these topics. So in the rough outline too, be able to discuss the factors that influence microbial death rates. 3, be able to discuss the 3 main ways which antimicrobial agents kill or inhibit cells. 4, understand the various types of disinfectants and how they work. And 5, understand the relative resistance of the major microbial groups to antimicrobial agents. Any question about the major goals of the rough outline? Might blow this slide up. So we're going to talk about the rate of microbial death and bacterial populations. When they're exposed to a microbial agent, they tend to die at a constant logarithmic rate. This is true when they're heated or treated with antimicrobial chemicals. That means if we were to start with 1,000,000 cells and they were to die at a constant microbial logarithmic rate, then something like a minute later, you would have 90% of the population dying. And so you'd only have 100,000. And then at 2 minutes, you'd only have 10,000. 3 minutes, only 1,000. 4 minutes, only 100. 5 minutes, only 10 survivors. And then after 6 minutes of exposure, only 1 survivor out of 1,000,000. If we were to plot that, and then this would be the x-axis would be regular arithmetic versus time. We would have the log of the number of microbial survivors. Then you can see that the death rate follows a linear constant rate. If you were to actually plot it on an arithmetic schedule, it's not going to be a constant rate. It's going to be a, what do you call that? A logarithmic rate, exponential rate, something like that. And this is much easier of a equation to work with. It's simply y equals mx plus p than this equation. And so microbiologists like to plot it on a semi-log plot. Realize this is a semi-log because the number of cells on the y-axis is the log number. And then on the time, that's the regular arithmetic number. Any question about that? I guess we can leave that there. Yep. When we look at the microbial death rate, the effectiveness of antimicrobial treatment depends on a number of factors, such as the number of microbes present at the beginning of the treatment. So if you want to kill all cells and you're treating with a certain agent, depending on how many cells there are, you will need a longer period if there are more cells. So longer treatment period if there's more cells. There are environmental influences that can affect the microbial death rate, like the presence of organic matter can affect the microbial death rate. Hospital specimens, let me state, almost always have organic matter with them. An example would be somebody has an accident in a hospital and they soil the floor with fecal matter, lots of organic matter around. Somebody comes up with bleach, the bleach will be inactivated by organic matter. And so to kill all of the specimens with bleach, you have to use a lot more bleach than you would if you just had the specimens alone, the bacteria cells alone. Any question about any of that? The temperature of the treatment can affect the microbial death rate. Generally, the warmer the temperature, the better the death rate, or the better the treatment. And then the presence of biofilms can also affect the microbial death rate. If the biofilm is present, the death rate will be much less than if the biofilm is absent. Any question about any of that? The time of exposure will affect the death rate, obviously. The longer times of exposure are required to kill off endospores than vegetative cells, for example. And then if there's more cells, to begin with, you need a longer time of exposure to kill off all the cells. There are microbial characteristics that can also affect the microbial death rate. And we'll talk about that, the microbial characteristics, in the end of this lesson or near the end of this lesson. Any question about any of that? All right. So this slide is showing you some of the terminology that you need to know: sterilization, the destruction or removal of all forms of microbial life, including endospores, but not necessarily prions. It's very difficult to kill prions. You can kill them if you submit it to 1,000 degrees. And I don't know if that's 1,000 degrees Fahrenheit or 1,000 degrees Celsius. Doesn't really matter. It's extremely hot. And we're a little bit confused as to why a protein needs 1,000 degrees, because that temperature will just char the protein. Generally, most proteins, you simply bring them to a boiling temperature, the protein will, what's the word I'm looking for? Denature, and then it will be non-functional. With the prion, that doesn't seem to be the case. That you actually need to get them to 1,000 degrees to destroy the prion protein. There is an experimental treatment, and that is you can treat prion disease with a proteinase, an enzyme which digests protein, and that does seem to destroy the prions. Any question about any of that? Commercial sterilization is sufficient heat to kill endospores of Clostridium botulism. It's usually used in the canned food industry. Disinfection, the destruction of vegetative pathogens, not necessarily of endospores. So like when we disinfect a toilet with bleach, we're only killing the vegetative pathogens. Antisepsis is the destruction of vegetative pathogens on a living tissue. Generally, an antiseptic is less caustic and less harmful to the skin than a disinfectant, but it isn't always the case. For example, you can clean the toilet with bleach, and then we call it a disinfectant, but if somebody has necrotizing fasciitis, the bacterial skin-eating disease, at least one of the older treatments was to put it in gauze with bleach, because that was an effective treatment to killing necrotizing fasciitis. In which case, bleach was both used as an antiseptic and as a disinfectant. Degerming is the removal of microbes from a limited area, such as around an injection site. We call it degerming when you alcohol swab a region. It does not remove all microbes. It just reduces the number to safe levels. And usually it's mechanical removal by alcohol soaked swab. Sanitization is the treatment intended to lower microbial counts on eating and drinking utensils to save public health records. So, like when you are cleaning dishes in a restaurant, that would be using sanitization. Any question about any of that? All right, some more terms. A biocide or a germicide is the substance that kills microbes. The biocide usually does not kill endospores. A bactericide is a antimicrobial agent that kills bacteria. A fungicide kills fungi. A virucide inactivates viruses. Bacteriostasis is the inhibiting the growth of microbes. It may not kill the microbes. Once you remove a bacteriostatic agent, the growth of the microbes might resume. And we talked about this before, we can have an antibiotic, which is biocidal, meaning it kills the cells, and penicillin G works that way. Or you can have an antimicrobial agent, an antibiotic, which is bacteriostatic, and that is it stops the growth of the microbes, but does not kill the microbes. And then once you stop giving that antimicrobial agent, the bacteria may begin growing. What we're relying on is the host immune system, removing the non-growing bacteriostatic agent, removing the infection from the patient. Any question about any of that? Sepsis refers to microbial contamination. Asepsis is the absence of contamination by unwanted organisms. Aseptic technique is an important part of surgery to minimize contamination from the surgical instruments, the operating personnel, and the patient themselves, as well as the air. Meaning, if you're to operate on the skin here and you don't use aseptic technique, well, the skin has a lot of microbes, which would then cause an infection. Why aseptic surgery is important, because when they first were doing surgery, about 50% of the patients died, usually from surgical infection. And I'm talking about before aseptic surgery came into play. Let's talk about the action of microbial agents. Antimicrobial agents kill or inhibit cells by one of three ways. They can alter the membrane permeability. If you do damage to the plasma membrane, either the lipids or proteins, you can cause the cellular contents to escape from the cell and interfere with normal metabolism, meaning the cell will die once the cytoplasm leaves the cell. Polymyxin B is an antibiotic which puts holes in the membrane of a cell, and how it acts is by damaging the plasma membrane. You can also kill or inhibit cells by damaging intracellular proteins. If you denature proteins, especially enzymes, by targeting disulfide bridges or hydrogen bonds, this will kill the cell. If you were to cook the cell or just heat it up, you would denature the proteins and that would kill the cell. It is true that alcohol denatures cellular proteins, but alcohol also damages the cell membrane. Any question about any of that? And then the third way an antimicrobial agent can kill cells is to damage the nucleic acid. This results in mutations and often results in cell death. If you have enough mutations in a cell, the cell will die. That's how X-rays kill cells, damaging the nucleic acids. Any question about any of that? It's also how UV light kills cells. All right, there are physical methods of controlling microbial growth. You can use all of these physical methods to control microbes. You can apply heat, you can apply filtration, you can put the microbes at low temperature, you can put the microbes under high pressure, you can desiccate the microbes, you can use osmotic pressure to control the microbes, or you can use radiation. Any question about any of that? Let's talk about heat. Heat is a physical method which kills microorganisms by denaturing proteins and enzymes. Though if you have high enough heat, such as flaming or incineration, you actually turn cells into char, and that will kill cells too. There are two types of heat. wet heat and dry heat. Wet heat includes boiling, autoclaving, and applying steam. Dry heat includes baking, putting things in an oven, and then flaming and incineration. Very high heat, these flaming and incineration. Moist heat denatures proteins by coagulating them, and obviously enzymes, which tend to be proteins. Boiling at 100 degrees C or applying flowing steam kills vegetative bacterial pathogens, most viruses, fungi, fungal spores, within 10 minutes and often much faster. Like you apply 100 degrees C to E. coli, the cells die almost immediately. Endospores and some viruses take longer to destroy. Some can survive 20 hours in boiling water. So some endospores are highly resistant to boiling heat. Any question about any of that? So how do we obtain reliable sterilization by moist heat? We use it in the lab if you were to come into the microbiology lab, and that is we apply temperatures above boiling. How was that achieved? We use a autoclave, which uses steam under pressure. It's very effective at killing vegetative cells as well as endospores. When you have steam under pressure, it raises the boiling point of water. And usually when we have an autoclave, we use 15 pounds a square inch. And the temperature is 121 degrees Celsius usually. And this causes sterilization within about 15 minutes. All organisms, including endospores, at least in 1 liter, are killed in 15 minutes. Any question about that? You can also sterilize the surface by applying steam directly, but the steam must contact the microbe directly. So something below the surface, like let's say you're steaming a clock and it's a real big clock, and you just steam the surface, you're really only killing the microbes on the outside of the clock. Anything inside the clock, especially far inside the clock, is not going to have the steam contact it, and the microbes will not be killed. So steaming usually only kills the microbes on the surface. Any question about any of that? All right, let's talk once more about pasteurization, a process discovered by Louis Pasteur. You don't need to know when, but you should know that Louis Pasteur discovered pasteurization. I mean, it's pretty obvious it's named after him. This reduces the number of spoilage organisms and kills all human pathogens. It is mild heating sufficient to kill organisms that cause spoilage, but does not kill all microbes, meaning pasteurized milk is not sterile. It does have microbes in it, and that's why it has a shelf life. How it works is it denatures proteins and enzymes, but the thermoduric organisms, which are heat resistant, can survive pasteurization. There are 2 treatments for treating largely milk, but any substance with pasteurization, and that is you can apply it for 63 degrees Celsius for 30 minutes, or you can apply it at 72 degrees Celsius for 15 seconds. Most milk that is pasteurized is applied for the second. And that's true of most beer as well. Beer that's pasteurized, most of it is treated at 72 degrees Celsius for 15 seconds. That's pasteurization. You can also treat milk at 140 degrees Celsius for around 1 second. This is not pasteurized milk, it's sterilized milk, and it's called ultra high temperature milk, or UHT milk. This type of milk does not require refrigeration till you open it. And sometimes you see ultra high temperature milk in the store. Sorry, what did I say? Score in the store. I think, what do you call it? Canned milk is UHT milk. And sometimes you can find it like a carton of milk, which is not refrigerated and it's sterilized, and then they sell it at room temperature. Now you have to be careful with that because sometimes they will irradiate it, and that's not ultra high temperature milk. If it's irradiated, it's also sterilized and can be kept at room temperature. Any question about any of that? So you could have different types of milk. You can have unpasteurized raw milk, which is not sold in the state of Washington and Oregon, so I don't know where you would find it, but that would have microbes from the cow in it, including human pathogens. You can have pasteurized milk, which most of it will be treated at 72 degrees Celsius for 15 minutes, although some of it could be treated at 63 degrees Celsius for 30 minutes. And then you can have ultra high temperature milk, which is sterilized milk. Any question about any of that? All right, let's talk a little bit about dry heat. We've talked enough about wet heat, so let's talk about dry heat. This cat is showing you that dry heat is not as efficient at killing microbes as wet heat, meaning the cat has climbed into the oven because the kitty likes it nice and warm, and obviously the cat can stay there for a while and obviously will not get cooked. Obviously, this oven is open, so the oven will cool down. But my point is, we all do this. We put our hands into an oven to pull out what we're cooking, and that does not burn us. You cannot put your hand through steam for any length of time without it burning you. So wet heat is much more efficient at killing microbes than dry heat, at least baking. Any question about any of that? So dry heat kills cells by oxidation. Hot air sterilization, that we normally call baking. Items are placed in an oven for at 107 degrees Celsius for 2 hours. That's much longer than we have to put microbes in boiling water to kill most microbes. Now, other examples of dry heat are flaming or incineration, and these kill cells right away, but these are much hotter temperature. Flaming will be something like close to 1,000 degrees, not 170 degrees Celsius. Any question about any of that? All right, another physical method for controlling microbial growth is filtration. Filtration can be used on liquids or gases. What you do is you have a membrane and then you filter what you want to sterilize through the membrane. The membrane has pores in it that allow the gas or the liquid to pass, but not the microbes to pass. It's useful for sterilizing liquids, especially liquids that would be altered by heating, and some juices are filtered just to preserve the flavor, the taste, and vitamins. Whereas if you were to well, even pasteurize it, but ultra high temperature of the juice, you can affect the taste as well as the vitamins. Coors beer is actually filtered. Why Coors beers taste different than most other beers is most other beers are pasteurized. Coors beer is filtered, and so it tastes a little different. Vaccines are also something which are frequently filtered. Milk is usually not filtered because the proteins and the fat in milk will clog the pores of the filter. Any question about filtration? All right, you can control microbial growth by putting them in low temperature. Low temperature inhibits the microbial growth. It usually does not kill the cells. Refrigeration is 0 to 7 degrees Celsius. It slows the metabolic rate of most microbes. Most microbes cannot reproduce or synthesize toxins at refrigeration temperature. However, there are some microbes, the psychotrophs, which can reproduce and grow at refrigeration temperature. And they can grow 3 times a day, meaning divide 3 times a day. And within a week, one or a few cells can then reach numbers greater than 2 million. And that is why food that has been refrigerated for a week, after that point, you should throw it away unless it has other preservatives in it. Food that's been refrigerated for about a week should be thrown away because there are psychotrophs in the refrigerator and they will grow in the food. Any question about any of that? All right. Lower temperatures, less than 0 degrees Celsius, results in freezing. This will stop metabolic activity. Slow freezing in your home freezer does allow ice crystals to form, which can kill some bacteria, about 50% of bacteria, and all parasitic species, meaning all eukaryote parasites, will be killed by slow freezing. Slow freezing is your home freezer. Fast freezing would be freezing things in liquid nitrogen. So you have it at like room temperature and then you expose it to liquid nitrogen. That fast freezing will freeze the water so quickly that ice crystals do not have time to form. And that will not kill most microbes. So most liquid nitrogen would not kill any bacteria. But slow freezing kills about half the cells, half the bacteria. Depending on the species, almost all bacteria will have some bacteria surviving, but like I said, there will be only about 50% surviving. You can also control microbial growth by putting the microbes under high pressure. High pressure transfers the pressure evenly throughout a liquid, and this will kill vegetative bacterial cells. The high pressure denatures proteins and alters carbohydrates. Endospores can survive high pressure. We can use high pressure to preserve the color, the flavor, and the nutrient value of juices. So many juices actually are preserved by high pressure. Any question about any of that? And here's a little joke about high pressure. The mama piece of coal says to the youngling piece of coal, your daddy's been under a lot of pressure lately. The piece of coal has been converted to a diamond. All right. Desiccation and osmotic pressure can control microbial growth. Desiccation is drying, removing the water from something that will prevent microbial metabolism. Cells can, however, remain viable for years. Endospores can survive desiccation for years, decades. They've even pulled endospores out of an Egyptian mummy, something like 8,000 years old, and the endospore germinated and then grew. I think the oldest endospore that they have recorded that survived in the Arctic ice. What they did was they took a core sample of Arctic ice. I should say Antarctic ice, not Arctic ice, Antarctic ice. And then so they drilled this core and then brought it up. And in the middle of the core, they found an endospore, and it was something like 40,000 years old. And they got it to germinate and then grow. Resistance to desiccation varies by species. Something like E. coli is very sensitive to desiccation and will dry fairly rapidly in desiccation, meaning under a day. And then obviously endospores can survive for years with desiccation. Some examples of desiccation including dried fruit, dried figs, high osmotic pressure like molasses and honey. These things don't spoil, molasses and honey, because of high osmotic pressure. A microbe falls into the molasses like E. coli, the water leaves E. coli, and then the E. coli dies. An endospore falls into molasses. It does not kill the endospore, but the endospore cannot grow. Cannot grow in the molasses or in the honey. Any question about any of that? So high osmotic pressure kills cells by the high salt or the high sugar, causing water to leave the cell, resulting in plasmolysis, where the plasma membrane shrinks and pulls away from the cell wall as water leaves the cell. That will kill many vegetative cells, but it will not kill endospores. Radiation is another physical method which can be used to kill cell. Radiation damages cells by damaging DNA. Ionizing radiation, that's X-rays, gamma rays, and electron beams, have a short wavelength. And the radiation ionizes molecules in water, producing ions, meaning an electron absorbs that radiation, gives that electron so much energy that that electron leaves the atom, leaving behind a charged molecule or atom, and that's why we call it an ion. These ions then can react with organic molecules, especially DNA, damaging the DNA. We use ionizing radiation to sterilize some food, some pharmaceuticals, and some medical supplies. Non-ionizing radiation, there's one that can sterilize or kill microbes, and that's UV light or ultraviolet light. How UV light damages cells: when UV light hits DNA, if the DNA have two thymines right next to each other, the UV light causes the thymines to form a thymine dimer, where the two thymines actually have two covalent bonds between them. Remember, two adjacent thymines do not normally bond to each other. The thymine diming causes the DNA to buckle, shown here, and that will not allow the DNA to replicate this part of the DNA, at least correctly, or will this part of the DNA be transcribed. Microwave radiation is not particularly antimicrobial, but microwaves do tend to heat up an object, and the heat can kill the microbe. I could mention that visible light is a form of non-ionizing radiation, but visible light does not kill cells. All right, this table gives you a summary of the different terms. I'll let you read through that. And then let's move on to the chemical methods of microbial control. When talking about the chemical methods, you should realize that few agents achieve 100% sterility. Most antimicrobial agents, chemical agents, decrease microbial numbers to safe levels or remove vegetative forms of a pathogen from an object. Most chemical antimicrobial agents do not kill endospores. There are a few that do, and we'll talk about them, but most chemical agents do not kill endospores. All right, so the principles of effective disinfection. Disinfection refers to the destruction of all vegetative pathogens. So disinfection does not refer to the removal of all endospores. Factors related to effective disinfection are one, the concentration of the disinfectant. Generally, the more concentrated the disinfectant, the better the cell killing. The time can affect the disinfection, and that is you need to have access to the surface. The disinfectant has to access the surface of the cell and then has to be applied long enough to disinfect or kill all cells. The pH can affect the disinfection. Generally, the more acidic the pH, the greater the cell killing. The presence of organic matter can also affect the disinfection. Remember, organic matter is often found in clinical situations. Organic matter tends to deactivate many disinfectants. Like bleach can be inactivated by organic matter. And so if you're cleaning with bleach or sterilizing with bleach, you have to use a lot of bleach when you run into organic matter before the bleach will kill the microbes. Any question about any of that? So generally with disinfection, you want it to be efficient, so you don't want organic matter around. There are different types of disinfectants. There are phenol and phenolics, biophenols, which are essentially phenols, bioguanides, halogens, alcohols, heavy metals, surfactants, surface active agents, chemical food preservatives, antibiotics, aldehyde, gaseous chemosterilizers, and peroxygens. Peroxygens are oxidizing agents. Many of these chemical disinfectants do not kill endospores. It is true that peroxygens like hydrogen peroxide and gaseous chemosterilizers, and then one of the others here, it might be a phenol, I don't remember, can kill endospores, but most do not. Like bleach is a halogen, it does not kill all endospores. Tincture of iodine is a halogen, it does not kill endospores. Alcohol is a chemical disinfectant, it's a weak one. It does not kill endospores. Heavy metals do not kill endospores. As far as I know, surface active agents, all of them do not kill endospores. Food preservatives do not kill endospores. antibiotics do not kill endospores. Any question about any of that? All right, this table is going through a summary of the chemical agents. I'm not going to go over it because we're running short on time. You only need to know the first three columns of the table. Let me blow this up. So you should know the type of chemical agent is, like all of the phenols are phenol or phenolic. You don't really need to know the mechanism of action. And then the preferred use, most of them are disinfectants. Yeah, you don't really need to know the mechanism of action. I'm not going to question you, like, is it bioguanine, disruption of plasma membrane? Let me talk a little bit about halogens because there are two very common halogens, and that is bleach and then iodine. They're both halogens. Iodine actually inhibits protein function and is a strong oxidizing agent. Chlorine bleach is a strong oxidizing agent. Iodine is usually used as an antiseptic, bleach usually as a disinfectant. Alcohol is a disinfectant, but it's very weak. And so we usually use this as a germicide, meaning this is something that we apply to the skin. It doesn't sterilize or really clean the skin. It just reduces the number of microbes to safe levels. Let me talk briefly about heavy metals. Sorry, my throat is getting very, what's the word? Coarse. Heavy metals were something that were used in the past, and then people stopped using them because better agents became available. The trouble with heavy metals is they are toxic, and they're fairly toxic to us, but they are effective against microbes, and their use has come back because of the development of antibiotic resistant microbes. And so, like when we use surgical dressing, they'll often put a heavy metal in the dressing to prevent the wound from getting infected. Any question about any of that? So this is one that was used in the past, and then better things became available. And so people stopped using it because, like I said, heavy metal, you don't want to consume this because it's fairly toxic to humans. Surface active agent includes soap, sanitizers, and then modern surface active agents used in hospital to clean the skin, used for skin instruments, utensils, rubber goods, things that are not susceptible to sterilization to heat, things that could be destroyed by other treatments. Probably won't talk much about food preservatives, but nitrates and nitrites are used in a lot of food, especially meat products, to help preserve those foods. And there are other chemical food preservatives. The trouble with chemical sterilizers, like ethylene oxide, is a gaseous sterilizing agent. It's very good at killing microbes, although it does take something like exposure to 15 minutes of the ethylene oxide gas. But the trouble is ethylene oxide is highly toxic to people, so you cannot use it where somebody would be exposed to it. It is useful for sterilizing medical implants, something that cannot be subject to heat. And then let me talk briefly about hydrogen peroxide and other peroxygens, killing cells by oxidation. These are very effective against endospores. So endospores can be killed by hydrogen peroxide, and they can also be killed by ethylene oxide, although endospores need a fairly long exposure. We're talking 15 minutes to half an hour to kill endospores by ethylene oxide. You just need exposure to hydrogen peroxide. So within a minute or two, the endospore would be killed by hydrogen peroxide. Comparing the effectiveness of various antiseptics, the exposure to soap and water, even after washing for 100 seconds, that's over a minute and a half, which nobody washes their hand for a minute and a half, except for maybe surgeons. I'm not sure how long they do, but they wash their hands for a long period of time. You'll note that soap and water only kills about 10% for 100 seconds of the microbes. So soap and water is not very efficient at killing microbes. The main benefit of soap and water is that it just washes away the microbes, doesn't kill them, and then any microbes that are left, only about 10% of them will be killed after exposure to 100 seconds. And normally, people wash their hands, even if they're doing a good job, only for 20 to 30 seconds. And so soap and water, very little killing. Tincture of iodine, that's iodine and 70% ethanol, kills over 90% of the cells after 100 seconds. And tincture of iodine used to be used very often before surgery to sterilize the skin. You would apply it to the region where you're going to be have the surgery, and then the surgeon could see where the iodine was to make sure that, you know, he's only cutting where the skin had been treated. And then iodine was not detrimental to the skin, so it could be applied and then applied for a long period of time. So it would have a long period to act. Actually, iodine is not used anymore before surgery, or I should say it's used seldom. And that is they've come up with even better agents than tincture of iodine. Any question about any of that? All right, so let's talk about some of the microbial characteristics and controlling microbial control. The most resistant agent to an antimicrobial agent are prions. To effectively kill prions, like I said, you need to apply temperatures of 1,000 degrees. And I don't know if that's Celsius or Fahrenheit. It doesn't matter. It's really high temperature where you convert the protein to char, and obviously the cells to prion, the cells to char too. Endospores of bacteria is the next most resistant microbe. Only a few treatment like hydrogen peroxide, ethylene oxide, and then one of the other chemical agents can kill endospores. It is true that radiation, long-term exposure to radiation, can kill endospores. Mycobacteria have mycolic acid in the cell wall, and so that makes many chemical antimicrobial agents unable to get through the cell wall. And so mycobacteria are resistant to many antimicrobial agents. Cystiprotozoa, the next vegetative protozoa. Gram-negative bacteria, the next most resistant. Gram-negative bacteria are more resistant than gram-positive bacteria. The gram-positive bacteria are more sensitive because they have an outer membrane of their cell wall, as well as the peptidoglycan is thinner, making the cells more sensitive to treatment. You can dry out the outer membrane of the cell wall, killing the gram. Oops, excuse me, I got that backwards. The gram-negative cells are more resistant because of the outer membrane of the cell wall than the gram-positive bacteria. The outer membrane of the cell wall can prevent the chemical disinfectant from getting in the cell, making the gram-negative potentially more resistant. And it really depends on what agent we're talking about. But the point is, gram-negative are more resistant than gram-positive. Off the top of my head, I know that penicillin G is an antibiotic that cannot get through the gram-negative cell wall, and it can get through the gram-positive cell wall and then damage the cell wall of the gram-positive bacteria. Fungi and fungal spores, the next most resistant. Viruses without envelopes, the next most resistant. Then there are more resistant than viruses with lipid envelopes. Gram-positive bacteria, the next most resistant. So this is second least resistant. And then the least resistant thing, which is the easiest to inactivate or kill, is a virus with a lipid envelope. You damage the lipid envelope or anything else of the virus, you can damage the virus. This is a simplified hierarchy of the relative resistance of the microbial groups to most of the chemical biocides. You should realize that specific agents can act differently on specific microbes, depending on the characteristics of the cells. For example, we state that the mycobacteria are fairly resistant. The only bacteria that is more resistant than the mycobacteria to most chemical disinfectants are the endospores, because of the mycolic acid in the mycobacteria not allowing the antimicrobial agent to get through the cell wall. However, alcohol, which is a very poor disinfectant, is actually very effective at killing mycobacteria. Why? Because alcohol can get right through mycolic acid. And then the mycobacteria can be easily killed by alcohol. So this cell, which is normally very resistant, is very sensitive to alcohol. And that is a specific characteristic of mycobacteria. Any question about any of that? I think I've already talked about that. I should mention that the reason why agents can get through the gram-negative cell wall is that the agent can move through the porin. If the porin selectively inhibits that agent will not get through the outer membrane. It will not kill the bacteria. So there's very few biocides which are effective on endospores, but there are a few, like all of the peroxygens, all of the gaseous disinfectants, which I call that something else. What did I call that? Gaseous chemosterilizers. All right, I've talked about viruses. And the main difference is the envelope of the envelope viruses. So the non-envelope viruses are more resistant because they only have a protein coat and then the nucleic acids, and you have to damage one of those two to damage the virus. Whereas the envelope virus, you can damage any of the envelope or the protein coat or the nucleic acid, so that the envelope viruses are more susceptible. I've already talked a little bit about prions, and that is they're very difficult to treat. You need incineration. How to treat prions is not completely solved yet, but you can treat them by incinerating at 1,000 degrees. And then for like surgical instruments that may have been exposed to a prion, you can treat them by proteinases. Expose the surgical instruments that cannot be exposed to 100 degrees Fahrenheit. You can expose them to proteinases, and that will get rid of the prions. All right. While microbial control methods are effective, they, especially the biocides, are not uniformly effective against all microbes. The compounds discussed in this lecture are not generally useful in disease treatment, with the exception of the antibiotics, meaning you cannot give bleach to a patient and then hope for the give it internally to the patient and then hope that that will remove the antimicrobial agent, because most of these agents would be toxic if consumed by a patient. The exception is antibiotics. Any question about this lesson? All right. If there's no questions about the lesson, that's it. I will see you on Thursday for a review. If you have questions about the lab, I'll be there from 6:30 to 6:45. All right, I'm going to say goodbye. Yeah,
Can I have just one favor? Is it possible to get the PowerPoints for week 8, week 9, and today?
yes.
That's the 13, 14, and 7.
Thank you for the reminder. Yes.
Thank you.
All right.
Bye-bye.
Thank you.
All right.